This video is continuation of video number 29, B part. In last video, that is the A part, we have learned up to exonucleases. Today we shall see the endonucleases. Restriction enzymes are called nucleases. They are of two types, exonucleases and endonucleases. We have already seen exonucleases. Now we shall see endonucleases. Endonucleases are a type of nuclease enzymes which remove or cut the nucleotides at specific position in the middle of DNA and produce sticky ends on each strand. It means endonucleases cut the DNA molecule in the middle of the DNA. Function. How these function endonucleases? Let us see. Restriction endonuclease first inspect the length of DNA molecule and identify a recognition sequence on DNA. Once the enzyme identifies the recognition sequence, then binds to the recognition sequence and cut two strands of DNA at specific points in sugar phosphate backbones. These enzymes are called restriction endonucleases because these enzymes cut the DNA molecule at specific or particular points. Okay. Now how the RN function? Now we shall see. Restriction endonuclease recognizes specific palindromic DNA. Then it cuts the DNA molecule. Palindromic DNA. What is palindromic DNA? Palindromic DNA is a type of DNA sequence that reads same nucleotides sequences into strands in forward and backward direction. Palindromic DNA reads the same nucleotide sequence in two strands in forward and backward direction. Look here, one example is there. The word Madam is there. The word Malayalam is there. Madam. We may read this from left to right. M-A-D-A-M. Madam. From right to left. M-A-D-A-M. Madam. So, the word may be read in both the directions. Similarly, there is one palindromic DNA molecule See the diagram. One strand of DNA having 5' GAATTC 3 dash. The opposite complementary strand having 5' 3 dash GAATTC. This is palindromic DNA. Okay. Restriction endonucleases cut the palindromic DNA between the two bases and opposite strands. See, there is arrow mark in the upper strand and in the lower strand. Now we shall see this example. Say, take for example, the enzyme ECOR first cut the palindromic DNA between G and A nucleotides. Produce single strand at the end. And these ends are called sticky ends or sticky points. Named so because at this region, hydrogen bond is formed with complementary strand. Hence, it is called sticky end. So, the very important point is the restriction endonuclease enzyme ECOR first cut between G and A nucleotides of palindromic DNA. Okay, this is how it cuts the DNA molecule. What is palindromic DNA? Very important for the examination. Now look here. Palindromic DNA is there. When it cuts between G and A, it produces in the upper strand AATTCG. In the lower strand AATTGC. So this is how the enzyme functions. Restriction endonuclease. For example, ECOR first cut between G and A nucleotides. Very important for the NEET. Hint third, cut between A and A nucleotides. It is specific. Okay. Now, shall pass on to next one. Restriction endonucleases are used in genetic engineering to produce recombinant DNA or rDNA. The recombinant DNA, as the name indicates, it consists of two DNA molecules. That is why it is called recombinant DNA. Okay, next. Ligases. The enzyme ligases are commonly known as joining enzyme. These enzymes are also known as molecular stitcher or glue because this enzyme joins two DNA fragments or pieces. What is the function of ligase? Ligase joins two DNA molecule. About this enzyme, you have already learned in the chapter Molecular Basis of Inheritance when we discussed about synthesis of DNA, where we come across ligase enzyme which joins two Okazaki fragments of DNA. Okay, what is the function of DNA? Ligases join two DNA molecules. The ligase obtained from E. coli bacterium and discovered by H.G. Corona, Indian scientist, but now is settled in America. Okay. Next, polymerases. Polymerases, DNA polymerase. This is also called DNA dependent DNA polymerase. This enzyme synthesizes DNA molecule. One more enzyme is there called reverse transcriptase enzyme. From this enzyme, a complementary DNA is produced. From this enzyme, cDNA is synthesized. How? Look here now. A brief explanation will be given. Now, the mRNA is produced during transcription. If such an mRNA is treated with reverse transcriptase enzyme, it produces single-stranded DNA molecule. If we treat this single-stranded DNA molecule with DNA polymerase enzyme, DNA molecule is synthesized. This is useful in genetic engineering. Okay, so that's all about this video. Video number 29. Thank you.